Hello students, so now let's discuss an important topic that how do we choose an antimicrobial drug? What are the properties, whether they are bacteriostatic, what is bactericidal, and how resistance is developing against antimicrobial. So in this topic, we will discuss various tables and various lists that will help you solve a question directly. So let's start with the first list of antimicrobial which are safe in G6PD deficiency. So the first table is telling you about the list of antibiotics which are safe in G6PD deficiency. So beta, what happens in G6PD deficiency? Inside RBC, red blood cells, there is an enzyme present that is G6PD. The function of G6PD enzyme is to make NADPH. And if this enzyme is genetically deficient, can we say NADPH levels will be very much reduced? And because of this reason, one antioxidant, that is glutathione, it will not be produced. Now, glutathione protects RBC from free radical damage, and any drug which can make free radicals in G6PD deficiency can cause RBC damage. And if RBC will become damaged, it can provoke hemolysis. So first point, which antibiotics are safe in G6PD deficiency? Remember a mnemonic PCM or paracetamol. So P for penicillins, C for the brother of penicillins, cephalosporins, and M for macrolides. Macrolides like erythromycin and azithromycin. Now, which antibiotics are unsafe? Unsafe means because they can provoke hemolysis in G6PD deficiency. So, to remember them, remember one rule. Any antibiotic which contains nitro or sulfa in it, they can cause hemolysis in G6PD deficiency because nitro and sulfa, they contain free radicals. And now glutathione is not there to protect this RBC from this free radical damage. Let's make a mnemonic for it. MAD, FAN, SCAN. So the first M stands for metronidazole. We have discussed that metronidazole, it is a nitro-containing drug. It comes under a group of drug nitroimidazole. So don't give metronidazole. Then we have antimalarials. And try to remember the names of two antimalarial, that is primaquine and quinine. But which antimalarial is safe in G6PD deficiency? The MCQ is mefloquine. Mefloquine is safe. Then D for dapsone. How we can remember leprosy drug dapsone? Remember, son word is coming. It's a type of sulfonamide only. Then we have fluoroquinolones, ciproflox, levoflox, aspirin. Aspirin is also a sulfa-containing drug. Then we have any nitro-containing drug like nitrofurantoin, one antibiotic. Similarly, if I'll ask you, which antihypertensive drug is nitro-containing? You can write nitroprusside. Don't give in G6PD deficiency. Similarly, one drug for angina, nitrates. Don't give in G6PD deficiency. Then we have sulfonamide. Obviously, they are sulfa-containing drug. And last drug avoided in G6PD deficiency is chloramphenicol. So now you have the first complete list. Drug should not be given in G6PD deficiency. Let's come to the next list now. So the next list is which antibiotics are safe in pregnancy. Again the same answer, PCM, paracetamol, that is penicillin, cephalosporins, and macrolides, erythromycin and azithromycin are safe in pregnancy. Now, which antibiotic are unsafe? Let's make a mnemonic. FATs. F for fluoroquinolones, A for aminoglycosides, T for tetracyclines, and S for sulfonamide. Now, remember, sometimes instead of sulfonamide, you might get an option of cotrimoxazole. Because we have discussed cotrimoxazole is a kind of sulfonamide. So one question comes in our mind, sir, why they are unsafe? So let's encircle first fluoroquinolone and tetracycline. Remember, fluoroquinolone and tetracyclines inside the body, they chelate with calcium. Because they chelate with calcium, what they will produce in fetus? In fetus, they can lead to bone and cartilage defect. Because bone and cartilage contain lot of calcium, so they will not be produced. Hence, they are avoided or not given in pregnancy. So what about aminoglycosides like streptomycin, gentamicin? Aminoglycoside, they can cause fetal ototoxicity. And the last drug we have is sulfonamide. Sulfonamide, we have discussed, they displace bilirubin from albumin inside the blood. Bilirubin becomes free, and if bilirubin enters into the brain of fetus, it can cause brain damage. So in fetus, sulfonamides, they can provoke brain damage due to bilirubin, which we call it as kernicterus. So remember, fats should be avoided in pregnancy. So now let's come to the third table or third list. Which antibiotic are safe in renal failure? So antibiotics which are safe in renal failure or renal safe antibiotic, we can make a mnemonic: contraindicated in renal failure totally. But before writing their names, why they are safe in renal failure? The simple reason is that these antibiotics, they are not excreted in urine. That means they are not excreted in urine. They are rather excreted in bile through liver. So can we say if there is renal failure, we can safely give them? So one question comes that these antibiotics do not require any dose adjustment in renal failure. So they don't require any dose adjustment in renal failure. And can we say because they are excreted through liver in bile, these antibiotics are hepatotoxic and they must be avoided in liver disease. Avoid them in liver disease. So they are safe in renal disease but avoided in liver disease. So the mnemonic is contraindicated in renal failure totally. Renal failure disease totally. So C stands for 1. Cephalosporin cefoperazone. We have discussed in cephalosporin why it was safe in renal failure. Same reason. I and R for 2 anti-tubercular drug, isoniazid and rifampicin. They are safe in renal failure but they are hepatotoxic. Don't give in liver disease. E for erythromycin and A for azithromycin. So they are macrolide. The only penicillin which does not require any dose adjustment in renal failure is nafcillin. Now L for a very famous drug, very important, linezolid. F for fluoroquinolones. But in fluoroquinolones, remember only 3 fluoroquinolones are safe in renal disease, but they are hepatotoxic. PMT. P for pfloxacin, M for moxifloxacin, and recently asked question was trovafloxacin. Then a very important question, which tetracycline is safe in renal disease? That is doxycycline. And the big brother of doxycycline is tigecycline. So this list is important. Question comes very frequently from this list. Now let's come to the next list, that why we call certain antibiotics as bacteriostatic and why we call them as bactericidal. So just remember one point, that bacteriostatic, they will only produce growth suppression. They will not produce the complete killing of the bacteria. While bactericidal, they will produce complete killing. That means no bacteria will remain. So one question comes, sir, then why we use bacteriostatic? Remember, bacteriostatic antibiotic, they reduce the bacterial load. And now very less amount of bacteria are remaining. Remaining bacteria, they are killed by immunity of the body. So can we say if a patient is immunocompromised, in immunocompromised patient, the drug of choice will be bactericidal drugs. So if I write a state of immunocompromise, let's say febrile neutropenia. Can we say in febrile neutropenia, neutrophils are not working properly? There is less neutrophil count. It's an immunocompromised state. Would you give bacteriostatic or bactericidal drug? Yes, we will give bactericidal drugs. So let's understand why we call certain antibiotics as bacteriostatic, why we call them as bactericidal. So imagine in a microbiology lab, you have a culture plate or a broth in which bacterial growth is there. What you did, you started adding antibiotics slowly and slowly. So as you started adding antibiotics and increasing the concentration, don't you think after one concentration of antibiotic, you will not see any visible growth on agar plate? So can we say that after one concentration of antibiotic, you will not see any visible growth of these bacteria or microorganism? So this concentration of antibiotic in which there is no visible growth on agar plate, this is known as MIC, which stands for minimum inhibitory concentration. Now, there could be a possible reason that although we are not seeing any visible growth, but there is one possible scenario that if we are not seeing any visible growth on agar plate, and if we take this agar plate under a microscope, there might be some microscopic growth on this agar plate. So no visible growth, MIC, but yes, microscopic growth can be there. So what you did, you started increasing the concentration further. When we started increasing the concentration further, after one concentration of antibiotic, which is MBC, minimum bactericidal concentration, there will be no microscopic growth as well. Can we say if there is no microscopic growth, there is complete killing of the bacteria? So keeping this thing in mind, let's see what do you mean by the word bacteriostatic and bactericidal. So let's say on agar plate for 24 hours, we added concentration of an antibiotic. As we are increasing the concentration in MIC which stands for minimum inhibitory concentration of an antibiotic which produce no visible growth. After 24 hours, we call it as MIC. But there might be a chance there can be microscopic growth. So what we did, we started increasing the dose of antimicrobial further. And when there is no microscopic growth as well, can we say there is 100% killing of the bacteria? So this concentration is known as MBC, which stands for minimum bactericidal concentration. Now, which drugs are known as bacteriostatic? Bacteriostatic drugs are those drugs whose difference between MIC and MBC is very large. Can we say that in normal dose, they will only suppress the growth, MIC. But if you have to do complete killing, you have to give very high dose of bacteriostatic drug, which might be toxic in humans. But what are bactericidal drugs? Bactericidal drugs are those drugs whose MIC and MBC are very near to each other. So can we say there is not much gap? So these drugs, they produce complete killing of the bacteria in a normal dose range also. So high gap in MIC and MBC, bacteriostatic. Low gap or almost same concentration, bactericidal. So let's make the list of drugs which are bactericidal, complete killing, bacteriostatic, which will only suppress the growth. So for bactericidal, let's make a mnemonic, Bewafa Meri Sanam. BE stands for beta-lactams. So in beta-lactams, we discussed 4 drugs, that is penicillins, cephalosporins, Carbapenems, and Monobactam. So among them, try to remember penicillins and cephalosporin, they are bactericidal drug. VA stands for vancomycin. Vancomycin is a bactericidal drug. F for fluoroquinolones. These drugs, they end with the word floxacin, ciprofloxacin, levofloxacin. So can we say if a patient is immunocompromised, febrile neutropenia, we should give beta-lactams, we should give fluoroquinolones, etc. A for aminoglycosides. So aminoglycosides are the drugs like streptomycin, gentamicin, etc. Now M for metronidazole and S for streptogramin. So these are the drugs which are bactericidal drug. But yes, if you want to add one more drug just for the reference, just write one drug, colistin. Colistin is also a bactericidal drug. So very commonly asked question. Now let's talk about the list of bacteriostatic drug. Mnemonic is MS Colt. MS stand for macrolides. So macrolides are erythromycin, azithromycin, sulfonamides, they start with the word sulfa. C for chloramphenicol. O for, we can write linezolid. So linezolid comes under a group oxazolidinone. So both will have zolid word in them. Then we have lincosamide. In lincosamide, we have a group that is clindamycin. And lastly, tetracycline group. So in tetracycline, we have doxycycline, minocycline. All these drugs, they end with the word cycline. So now let's come to the next list. How do we choose the dose of an antibiotic? So let's understand one point. Let's say a patient is suffering from pneumonia. So don't you think we need some concentration of antibiotic which should reach to the lungs? We should reach to the lungs and kill this pathogen or microbe in the lung. Now, unfortunately, we cannot calculate what is this optimal dose, that how much of this antibiotic is reaching to the site of infection. No. But what we can do, we can only check the concentration of antibiotic in our blood because we can only take blood sample. But we have discussed from the previous section, in blood, at least we have to maintain MIC of any antibiotic, that is minimum inhibitory concentration, which we have found from agar plate. So all antibiotics, what we do, we maintain at least MIC in our plasma or in our blood. Now, how much antibiotic we must give above MIC? Because MIC is the minimum inhibitory concentration in our blood. Now, let's see, there are certain antibiotics which require very high concentration above MIC, minimum inhibitory concentration. And when they are given at a very high concentration above MIC, they show rapid killing of bacteria at the site of organ. So this is known as CDK. CDK stands for concentration dependent killing. That means we have to give very high dose above MIC, then they will produce rapid killing of the bacteria. So certain antibiotics, they don't show CDK, concentration dependent killing. Certain antibiotics, they show time dependent killing. Time dependent means they don't require very high concentration above MIC. No. What they require is prolonged exposure or repetitive doses. So if they are given for prolonged duration of time, they show rapid killing of the bacteria. So if I'll ask you a question, among CDK and TDK, which antibiotic would you like to give once a day in a very high dose? That will be CDK. So concentration dependent killing, if any antibiotic shows, these antibiotics are given once a day. But if a drug shows time dependent killing, these drugs are given multiple times a day because we have to give for prolonged duration of time. Now, there are certain drugs which can show any of the following. That means they can show concentration dependent killing also. They can also show time dependent killing. So don't you think height into base, this is known as area of a triangle? So this CTDK, which is concentration and time dependent killing, both, it is also known as area dependent killing. And if a drug shows area dependent killing, don't you think we can give this drug both once a day and multiple times a day? But think on the perspective of a patient. Which will be convenient for the patient? Which will be cheaper for the patient? Yes, it once a day. So again, these CTDK, they are preferred to be given once a day. So this is three patterns of killing of an antimicrobial: concentration, time, and both area dependent killing. Now another concept is very important. Just imagine you have stopped giving an antimicrobial. Do you think if we stop giving an antimicrobial, its concentration falls below MIC? Now, do you think it will show any killing? Logically, if we say they will not kill a bacteria because their concentration is below MIC. But in our body, we have immunity. Immunity is not on agar plate. Our body has immunity. So even after stopping an antibiotic, killing can continue. So after stopping an antibiotic, killing can continue. And if killing can continue after stopping an antibiotic, this effect is known as PAE. PAE stands for post antibiotic effect. So post antibiotic effect means killing continues after brief exposure to an antibiotic because our immunity gets stimulated. Now the importance of PAE is that you don't have to give an antibiotic for very long duration of time. Let's take an example. When we suffer from sore throat, we take amoxicillin for 7 to 10 days. But we take azithromycin as well just for 3 days. Why amoxicillin for 10 days? Why azithromycin only for 3 days? Because I will be telling you that azithromycin shows long post-antibiotic effect. That even after stopping azithromycin just after 3 days, killing continues. So with post-antibiotic effect, we can reduce the total duration of therapy. So let's talk about these points. C was for concentration dependent killing. That means we must give very high concentration of antibiotic above MIC, and these antibiotics are given once a day. They are not given multiple times a day. Let's take an example. If we take an example, one aminoglycoside streptomycin, one another tuberculosis drug rifampicin. These antibiotics are given for the treatment of TB. What do you think? We give rifampicin once a day or multiple times a day? We give aminoglycoside streptomycin once a day or multiple times a day? In TB, we give these drugs only once a day because streptomycin and rifampicin shows concentration dependent killing. So time dependent killing, these drugs are given multiple times a day. So let's take an example like penicillins, amoxicillin, ampicillin, similarly cephalosporins like ceftriaxone, many cephalosporins you know. So just imagine when you suffer from sore throat or any person suffers from pneumonia. For pneumonia, we prescribe penicillins 3 times a day. We give ceftriaxone injection 3 to 4 times a day. Why? Because they show time-dependent killing. Now, CTDK is a combination of both concentration and time-dependent killing. It is also known as area-dependent killing. So, these drugs can be given either of the way. Now, what was post-antibiotic effect? Post-antibiotic effect is prolonged growth suppression of a bacteria prolong growth suppression of a bacteria after brief exposure to an antibiotic. So that means after stopping an antibiotic, the killing continues because immunity becomes stimulated. So let me give you an example. In the treatment of leprosy, we give rifampicin, and we give rifampicin once a month. We give rifampicin once a month in the treatment of leprosy. So don't you think it is too less to give once a month? The answer is yes. Rifampicin has prolonged or very long post-antibiotic effect. So if you give once a month, killing can continue for whole month. So rifampicin shows long post-antibiotic effect. So finally, we have understood the concept. Let's make the list of the drugs which show variety of this property. So the first property is CDK, which is concentration dependent killing and a very long post-antibiotic effect. And this is the list you have to remember for your exam. Most of the time the question comes from this list only. And the mnemonic is MARS, metronidazole, aminoglycoside. So in aminoglycoside, you will get an option of streptomycin, gentamicin, and rifampicin. These are the most commonly asked questions. S stands for streptogrammin. And among all lists, the longest post-antibiotic effect is of streptogrammin. Longest post-antibiotic effect. Then we have CTDK, that is concentration, time-dependent killing, which is also known as area-dependent killing. It is also known as area-dependent killing. And these drugs also have long post-antibiotic effect. So remember, any drug which shows concentration-dependent killing has long post-antibiotic effect. Three drugs are there: daptomycin, vancomycin, and fluoroquinolone. Try to remember vancomycin in it. And fluoroquinolone was also asked in one exam. Now certain drugs, they show time-dependent killing. They have to give multiple times a day. And they have long post-antibiotic effect. But certain drugs, they show time-dependent killing, and these are the only drugs which don't have very long post-antibiotic effect. So the drugs which show time-dependent killing and long PAE are ATT, azithromycin, its brother telithromycin, and tetracycline. So tetracycline, like doxycycline, it is given twice or thrice a day. So that is why we give multiple times a day time-dependent killing. Time-dependent killing with short or no post-antibiotic effect, CPEC. Try to remember clindamycin, penicillins, and cephalosporins. They are commonly asked questions, but the most important one is MARS. So now let's talk about the last part of this chapter, a very important part, upcoming problem, antimicrobial resistance. So in antimicrobial resistance, the first type of resistance is inherent resistance. Inherent means when we make this antimicrobial, this bacteria was never killed or never effective by that antimicrobial, because that antimicrobial does not have target to kill the bacteria. So, and these antimicrobial are never effective against that bacteria. So this is inherent resistant, and remember, it is not a problem for us. It is not a problem, because against these bacteria, we will not use this antibiotics. So let's take an example, like example, penicillin G. So penicillin G, it is not effective against Enterobacteriaceae. Enterobacteriaceae means E. coli, Klebsiella, Pseudomonas. Similarly, metronidazole. Metronidazole, it cannot kill any aerobic bacteria. It is only given against anaerobic bacteria. So we don't use metronidazole against aerobic bacteria. Similarly, aminoglycosides. Aminoglycosides, they cannot kill anaerobic bacteria, opposite to it. So we don't give aminoglycoside against anaerob. So this inherent resistance is not a problem. Don't use this antibiotic. What is the big problem is acquired resistance. Acquired resistance means with time, the sensitive bacteria is acquiring resistance. And this is a very big problem at today's time. We are getting out of option and bacteria and microbes are winning the war. So how does a bacteria acquires or develops a resistance against any antibiotic? Let's learn it. The first cause of resistance is bacteria. It has developed certain mechanism by which it is not allowing a drug to enter inside it. So drug influx is hampered. Second cause of resistance is, let's say this drug or antibiotic entered into the bacteria, but this bacteria made a special transporter which throws this drug out. So this is known as drug efflux. Third cause of resistance is, when this drug entered into the bacteria, bacteria made enzymes and these enzymes degrades this drug. And lastly, this is known as enzymatic inactivation. And lastly, when we gave a drug, let's say that drug kills a bacteria by certain mechanism, cell wall inhibitor, cell membrane inhibitor, protein synthesis inhibitor. Now the bacteria, its target is modified. There is mutation in the target. So this antibiotic cannot inhibit cell wall because cell wall is now of different type. So there is target modification. So let's see which drugs are developing resistance by these four mechanisms. So drug influx, that means the bacteria is not allowing a drug to enter. Resistance to carbapenems. Carbapenems are the drug like imipenem, meropenem. They are not able to kill nowadays Pseudomonas. Because Pseudomonas is not allowing a drug to enter inside them. So resistance in Pseudomonas against carbapenem is due to hampered drug influx. Now which antimicrobials are becoming resistant because of drug efflux? The bacteria is throwing out the drug. Remember a mnemonic ATT. ATT is anti-malarials like chloroquine. So chloroquine resistance in malaria is because of drug efflux, tetracycline and tigecycline. Now enzymatic inactivation, when the drug entered into the bacteria, bacteria made certain enzymes and these enzymes degrades this drug, the drug becomes inactive. The classical example are A, B, C. A for aminoglycosides, beta lactams like penicillin, cephalosporin. So there was an enzyme we discussed, beta lactamase enzyme, which degrades beta lactams, and C for chloramphenicol. So ABC is the cause of enzymatic inactivation. Lastly, mutation in the target. So let's say you develop penicillin and penicillin kills Staphylococcus aureus by inhibiting transpeptidase enzyme, which we learned. Now, if there is mutation in transpeptidase enzyme, can we say the target of penicillins? It is now different type. Penicillins cannot kill this type of Staphylococcus aureus. So the classical example of mutation in the target is MRSA, which stands for methicillin resistant Staphylococcus aureus. So there is mutation in transpeptidase enzyme, which you also call it as penicillin binding protein 2A. Similarly, resistance in macrolides and resistance in fluoroquinolones. So macrolides, they inhibit protein synthesis like erythromycin, azithromycin, which are macrolide. They inhibit protein synthesis by binding to 50S ribosome of the bacteria. So there is mutation in 50S ribosome. So can we say it cannot bind to its target? The target has been modified now. Similarly, fluoroquinolones, ciprofloxacin, levofloxacin, they kill a bacteria by blocking or inhibiting DNA gyrase enzyme. So if there is mutation in DNA gyrase enzyme, can we say the target of this antibiotics has been modified? They cannot bind to their target. This leads to resistance. So let's remember, let's make a mnemonic: drug efflux, ATT. enzymatic inactivation ABC, and target modification MMF. So these are commonly asked questions, and this finishes our choice of antimicrobial. Let's solve certain MCQs. So which of the following drugs should be avoided in third trimester of pregnancy? So I have told you 4 drugs which are unsafe in pregnancy, that is fats, fluoroquinolone, aminoglycosides, tetracyclines, and S for sulfonamide. And with sulfonamide, I have told you cotrimoxazole is a type of sulfonamide, because sulfonamides, they can cause kernicterus in fetus. Next question, which of the following drugs, if used alone, would primarily inhibit bacterial growth rather than killing the pathogen? Can we say the question is asking about which drug is bacteriostatic? Because bacteriostatic will only reduce the growth. Bactericidal will cause complete killing. So bactericidal, we have discussed a mnemonic Bewafa Meri Sanam, and bacteriostatic were MS Colt. So among them, remember azithromycin, which is a macrolide, is a bacteriostatic agent. So fluoroquinolone, vancomycin and ceftriaxone, they are bactericidal drug. So which of the following antibiotic exhibit concentration dependent killing, that is CDK, with long post antibiotic effect? So let's see which drug. Clindamycin beta we discussed, it shows time dependent killing with short PAE, short post antibiotic effect. Rifampicin shows concentration dependent killing, that is why it is given once a day in TB, and it shows long post antibiotic effect, once a month in leprosy. Vancomycin shows both CTDK, concentration and time dependent killing, which is also known as area dependent killing, with a long post antibiotic effect. And similarly with moxifloxacin or fluoroquinolone, CTDK and long PAE. So the correct answer for this question is rifampicin. So this finishes our chapter. Best wishes and thank you very much.